Yeah, we got 6. Okay, well, I'm going to start. First, remind you that Wednesday is a quiz, typical quiz. 10 points, 15 minutes, 2 attempts, and it covers molecular genetics, enzymes, and energy, and photosynthesis. Another thing, I'll send you an announcement about this, but you can see my tentative plans for the remainder of the semester if you go to the Canvas calendar. That schedule might change depending on circumstances, but that's the way it looks to me at the moment. Okay, we still only have 6 people, but I'm going to start going over the material, and hopefully more people will join. Can you see my screen overview?
Yeah, we can see it.
Okay. You've seen this before. These are the topics that we're going to either we have covered or we're going to cover between now and the end of the semester. We've already done energy and enzymes, and today I'm going to focus on photosynthesis. Then on Wednesday, respiration, how we get energy from food. The photosynthesis. All life on Earth requires a constant input of energy. Remember the second law of thermodynamics, that there's this condition called entropy, and all systems tend to go to maximum disorder, and to avoid that, you have to have an input of energy. And for biology, the energy comes from the sun. The producers of food are anything that's green, anything that's photosynthetic, and that's usually green plants. Animals are consumers. They never produce food. They can only eat plants or each other. And food chains always start with photosynthetic organisms. Now, I want to make one brief point here, and that is that there are alternatives to the sun for capturing energy for living things. And one of these is called chemosynthesis. So whereas the sun photosynthesis captures the sun's energy, deep in the ocean, there are organisms that don't need light. They can derive energy from carbon dioxide and water and hydrogen sulfide along with oxygen. So just bear that in mind, that there are these weird organisms. You see pictures of some of them here on the right. Tube worms. They are not green. They cannot produce food, but they can generate energy for themselves by chemosynthesis. So I'm going to do some review before getting into photosynthesis itself. The electromagnetic spectrum, how can we capture and make use of the energy that's available in the electromagnetic spectrum? And the answer is photosynthesis. Well, you remember this picture from last time. It shows the full spectrum of electromagnetic energy from very long radio waves to very short energetic gamma rays. This clear section shows the wavelengths of visible light, what we can see. And it turns out that plants can see pretty much the same wavelengths that we can see, with the exception that plants cannot see green wavelengths. They can see red, yellow, and blue. They cannot see green. And the reason is that chlorophyll can absorb red and blue, can't absorb green, and therefore it reflects it. And this shows the absorption spectrum of the two major forms of chlorophyll. This is a structure of a chlorophyll molecule. You'll notice it has a magnesium atom, the center of it. So this would be a cofactor that it is needed for its ability to absorb light. So both of the chlorophylls absorb blue light, 400 to 500. They absorb red light from 650 to 700, but they do not absorb green light. So as I said, they reflect it instead. So where in a plant does photosynthesis occur? Well, it occurs in any part of the plant that's green. So it can occur in the stems. Roots are rarely green, so photosynthesis doesn't occur in roots, but the major place is leaves. Plants that have leaves, which are usually broad, which gives them a lot of surface area to absorb light. So the leaf has a blade and a petiole. And if you take a little section out of the leaf and look at it under a microscope, you'll see that it has two sides, an epidermis on each side. The lower epidermis has little openings in it called stomates or stomata. These are for gas exchange. So the plants can give off oxygen and take up carbon dioxide. There's this layer of cells called the mesophyll. And if you look at just one of these mesophyll cells, you'll see that it has cell wall. There's a cell membrane, it's not shown here. Nucleus, a large vacuole, and these little green bodies called chloroplasts. So what is the structure of a chloroplast? So here we're going from this picture at the bottom of this slide, same picture at the top of this one. And now we're going to focus in on a chloroplast. A chloroplast has an outer membrane, an inner membrane, and then two regions that you want to remember. You haven't heard these terms before, but grana and stroma. So the grana are these stacks of membranes called thylakoids, and the stroma is the space between the thylakoids. And those are two critical regions for the capture of energy. Now, if we focus in on just a granum, it is a series of membrane-bound structures called thylakoids. So this is a thylakoid membrane. There's an interior space, and then exterior to it is called the stroma. And what's important about this is that photosynthesis involves two types of reaction called the light reaction and the dark reaction. The light reaction goes on in the grana, in particular within the thylakoid membrane. The dark reaction occurs in the stroma. It's called the Calvin cycle, and we'll talk more about that. Now, the thylakoid membrane is critical in photosynthesis. That's where all the action involving light occurs in the thylakoid. So remember thylakoid membrane, remember these points about it.
Professor, the thylakoid, that's like what when we cut the aloe vera plant, and we see that little stuff inside. Is that the same?
I don't think so.
Oh,
You
okay.
you need a microscope to see
that?
a
Okay.
thylakoid membrane. But that's where sunlight is captured. That's where there is an electron transport chain that we talked about before. Talk about it again today. That's where chemiosmosis occurs. And that's where ATP is produced for the dark reaction. And the dark reaction is where we get sugar. So remember these things about the chloroplast, in particular about the thylakoids. And now here's more review, redox reactions. Energy is harvested. Remember, this is what we want. We want to harvest energy from the sun through electrons that are moved from one molecule to another. These electrons carry energy, and they go on a downhill journey. And while they're doing that, they release energy. The basis for electron transfer is a series of reactions called redox, oxidation reduction reactions. Now, some substances more strongly attract electrons than others. A substance that loses an electron is said to be oxidized. And if it gains electrons, it's said to be reduced. Remember those two terms. Oxidized is loss of an electron, one or more. Reduced is gain. And these two processes always occur in pairs. If something is oxidized, something else is reduced. And you've seen this before. The most basic illustration of redox. So substance A is reduced because it has an extra electron. Substance B is oxidized. If A transfers its electron to B, A is now oxidized and B is reduced. On biological oxidation, the molecules we're concerned about are organic. So if we have an organic molecule that includes two extra hydrogen atoms, so for example, this could be glucose in a reduced form, and it these electrons, these hydrogen atoms, which in each one is a proton plus an electron, they're transferred to an electron carrier. In this case, NAD, it's a coenzyme. Now, the glucose has been oxidized, and the carrier is now NADH plus a proton. And it's now a reduced electron carrier. Now, I want to show you a video. And this video is actually about redox in mitochondrial membranes, which we're going to talk about in particular next time. But this is a good illustration of what goes on in membranes in general, including in photosynthesis. So let me stop sharing this and go to
all eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality, they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
You back with me? Biological oxidation. Anybody listening?
See, I'm back.
Yeah. Yeah.
Okay.
I'm listening to.
Yeah.
Oh,
listening too.
okay. Okay, well, I can't tell until you say something.
We're here.
So, okay, as I said before you saw the video, the video is about mitochondria, but it's really about membranes and about redox reactions within membranes. So I'm going to talk now about photophosphorylation and about electron carriers. Now, the video kept mentioning electron carriers, and that's what's illustrated here. So we have an A substance, which is reduced. It has extra hydrogen atoms. And when it's oxidized, it goes to A without, but the electrons are transferred to a carrier, NAD, and that becomes NADH and releases energy. Then the electrons are transferred again to another carrier, in this case B, and it becomes reduced, BH2. The important point here with regard to photosynthesis is that these electron transfers are made within the thylakoids. Remember that. This is a drawing of a chloroplast. Remember, there's stroma, where the dark reactions occur, and there are the grana, which are the stacks of thylakoids. That's where the light reactions occur. And so the electron transport chain and ATP synthesis goes on in the thylakoids. And here we have a visualization of that. Now, this should look very similar to you from what you saw in the video. The protein complexes in this membrane, and this is the thylakoid membrane. The protein complexes are not the same ones in respiration, in mitochondria. Nevertheless, they do all the same things. So if you start at the beginning here, an oxygen evolving complex, you see that light comes in, light energy comes in. The first thing that happens is that a water molecule is split into two so that there's molecular oxygen plus a proton plus H plus. And that the H pluses accumulate on the what's called the lumen side of the thylakoid membrane, so that there are more protons on the lumen side than on the stroma side. Remember, the stroma is out on the outside, the lumen is on the inside of the thylakoid membrane. That's what you see here. So the electrons are transferred, and as they lose energy, and protons continue to accumulate on one side of the membrane. And finally, they keep losing energy, but they arrive at another photosystem. It's another chlorophyll pigment. And again, this can accept light, and that energizes the electrons to go on and make more reducing power. More NADP is converted to NADPH. Now, these protons that keep accumulating as light comes in, keep accumulating within the thylakoid lumen. They eventually are passed through this ATP synthase complex, and this process is called chemiosmosis. And when that happens, ADP plus inorganic phosphate is converted to ATP. And this, of course, is the whole point of the exercise, to generate energy, starting with light, and generate chemical energy in the form of ATP, which then can be used to drive all the processes in the cell and keep the cell alive. Any questions about this light reaction, where light comes in to photosystem 2, it activates electrons, which are transferred through the chain. And as the electrons are transferred, protons accumulate on one side. And at the end, protons move through the membrane through this ATP synthase and provide the energy to convert ADP to ATP. This is another illustration. The next slide shows the same thing as this, except that it shows the activation energies. So here you have the sun emitting light. We have two photosystems, and this is all within the thylakoid membrane. So remember, here's the thylakoid membrane. And now light strikes chlorophyll with an absorption wavelength of 680. It's in photosystem 2. It excites electrons from low energy to high, and then they start to pass through various protein complexes within the membrane. These are the protein complexes within the membrane. And as they do, they lose energy. And finally, they get to this second pigment, P700, which is called photosystem 1, and absorb another quantum of light, again get excited, a high energy, and you can go down another electron transport chain through a series of proteins to NADPH. And at this point, the light reaction is over and the dark reaction starts. The dark reaction is called the Calvin cycle, and that's the part of the chloroplast that makes sugar, primarily glucose.
My followers help you, dog. You know how many views I get on my stories? You know how many views?
So
Actually,
stages of photosynthesis, could somebody mute their mic? I'm getting some feedback.
chicken one's good enough for me.
Anthony, is that your mic that's on?
Oh, probably.
Thank you. So here's back off from the fine points and look at the overview of what happens in photosynthesis. So you have the chloroplast here with the thylakoids. They are the critical part of the chloroplast. And what happens, sunlight comes in and provides energy. It splits water. As water goes in, it gets split, and out comes oxygen. But the electrons that are generated from the splitting of water molecules go into an electron transport chain through a series of carriers. And I just want to remind you that for each of those steps in a membrane, one of these reactions occurs. The handoff of electrons from one molecule to another, to a carrier, to another carrier, to another carrier. And I'll explain later why this is necessary. Why you can't just go from the beginning to the end in one step. You have to have all these intermediate steps. Okay, so that's the light reaction where water comes in and light comes in and oxygen comes out. Then also within the chloroplast, within the stroma, we have the dark reactions where carbon dioxide goes in and sugar comes out. And this is an amazing process. It's known as the Calvin cycle, and it's light independent. So the Calvin cycle is a set of chemical reactions that convert carbon dioxide to glucose. It uses energy from ATP and a carrier known as ADP, NADPH from the light reaction in a series of redox reactions to produce sugar in a stepwise process. So here's an illustration of the Calvin cycle at work. Carbon dioxide comes in, it's converted to 3 carbon sugars, which are then converted to 6 carbon sugars, primarily glucose. So the light reactions, to summarize this in words, the light reactions generate electrons. These are transferred via carriers, NADP, in the thylakoids, and that generates ATP transferred to the dark reactions of the Calvin cycle to make sugar. The Calvin cycle in more detail. Here you have the chloroplast with the light reactions and the dark reactions, Calvin cycle, and here's the Calvin cycle blown up. The first step is the truly amazing one, where a gas, carbon dioxide, 3 molecules of carbon dioxide, are taken in and fixed. Fixing means that they're converted into a chemical, an enzyme called Rubisco. So carbon fixation is a process by which plants convert atmospheric carbon from CO2 to form solid organic compounds, for example, glucose. And this process is done by this amazing enzyme complex called Rubisco. And here you see a 3-dimensional structure of Rubisco. And if you look at it up close, you see that there is a pocket which binds to CO2, carbon dioxide. So then there are a series of enzymatic reactions which finally lead to a compound called glyceraldehyde 3-phosphate. Here's a drawing of the structure of that. And you can see that it's a 3-carbon molecule. 2 of these put together make a 6-carbon molecule, and that is usually glucose. So Rubisco catalyzes the first step of this cycle, where it takes a gas from the atmosphere and converts it into energy-rich molecules like sugars, glucose. So stoichiometry of it, 1 turn of the cycle costs 9 ATP molecules and 6 energetic electrons. And these are obtained from the light reaction. So here you see it takes 6 ATPs to do this step, takes 3 more ATPs to do this series of steps here, plus more reducing agents, more reduced carriers, NADPH, in order to make the cycle go. And as the cycle goes, it keeps spitting out sugars. This is really doesn't say anything more except expressed somewhat differently from the previous slide. The point is the 3 molecules of carbon dioxide go in and 6 molecules of glyceraldehyde 3-phosphate come out at this point, and they are converted to 6 carbon molecules, usually glucose, and that's the high energy molecule for metabolism. That's what makes us go every day. So to restate the light reactions, oxygen is produced as a byproduct of the splitting of water. So water is H2O. You split a molecule of water, you get half of an oxygen molecule and a proton. And an electron. So the electron goes through the electron transport chain. The proton accumulates in the inner side of the membrane. And both of these are used then to make ATP. In the light reactions, the solar energy is converted to chemical energy in the form of ATP and NADPH. And these two molecules then are used to power the light-independent reactions, the so-called dark reactions, in photosynthesis. And both the light and the dark occur in the chloroplast. The dark reactions are also known as the Calvin cycle. In the Calvin cycle, carbon dioxide, a gas, is incorporated into sugars. It's called carbon fixation. And glucose is produced. If you hook a whole bunch of glucose molecules together, you get a polysaccharide, which is usually starch in a plant. A photosynthesis and respiration accomplish two completely different things. Photosynthesis makes food, respiration uses it. So photosynthesis, which is essentially the same as glucose synthesis, it's a metabolic pathway that is primarily anabolic, energy in. Where does the energy come from? It comes from the sun. So in photosynthesis, carbon dioxide and water are used to make glucose, C6H12O6, plus oxygen. When plants are in the light, they produce oxygen as well as sugar. Respiration reverses that. Respiration is glucose oxidation, and it is a metabolic pathway that is catabolic. That is, energy is produced. So if we take this same molecule, this green C6H12O6, here in respiration, plus oxygen are used to produce carbon dioxide and water. So when we breathe, we produce carbon dioxide, we get rid of it, and that's because glucose is being broken down to carbon dioxide and water, and energy is released. So,
Professor,
yeah,
before before continue, I was just wondering, are we gonna have to memorize any of the last formulas for photosynthesis and respiration?
memorize what?
The formulas, the little like C2.
Oh,
and all that?
going back. These items, these intermediates, no, you don't need to memorize those. You should know that, that the Calvin cycle produces glyceraldehyde 3-phosphate, and that it's a 3-carbon sugar converted to 6. And you should know what I've circled here, what Rubisco is and what carbon fixation is. You don't need to know anything about these protein complexes except that they carry electrons. And you need to know that when electrons are carried through the thylakoid, protons accumulate in the lumen on the inside. And that the end result of this long process is ATP synthesis, which is also known as chemiosmosis, where protons are pumped through the membrane from one side to the other. And when they do, ADP is converted to ATP. Those are the essential things that you'll need to know, and the terms that you'll need to know.
Okay, thank you.
Right, there are 3 ways that a cell can make ATP. And I'd like to just summarize those now. One is called substrate level phosphorylation. So you'll need to know these terms: substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. phosphorylation. So substrate level is direct enzymatic transfer of phosphate from a phosphorylated molecule to ADP in order to make ATP. Oxidative phosphorylation, this occurs in respiration, so we're not really talking about it today, but electrons from organic compounds like glucose are passed through a series of reactions, electron transport chain, to an inorganic molecule, usually oxygen. And then chemiosmosis generates ATP as energy is released. This happens in the mitochondria, not in the chloroplast. What happens in the chloroplast is photophosphorylation. Whereas pigments of photosynthetic cells, that's usually chlorophyll, trap the energy in light and convert it to chemical energy through, again, electron transport chain and chemiosmosis, generating ATP, which then is used in the dark reaction, the Calvin cycle, to make sugars. So I'm going back now and illustrating the three different ways that ATP can be made. The first, substrate level phosphorylation. It's a direct enzymatic transfer of phosphate from a phosphorylated molecule to ADP to make ATP. And this is a one-to-one ratio. So one molecule of the progenitor and ADP makes one molecule of ATP. So ADP is shown here. And phosphoenolpyruvate is the substrate shown here. This enzyme, pyruvate kinase, takes the two and makes one molecule of ATP and the product, another product. The important point here is that substrate level phosphorylation occurs, but it's not very productive. So pyruvate, which you see here, is nothing but glucose after it's split into three carbon molecules. So that's substrate level phosphorylation. We've just talked about photophosphorylation, which produces glucose in the chloroplast. The chlorophyll traps light energy and converts it to chemical energy through this sequence of electron transport plus chemiosmosis plus carbon dioxide fixation. The energy that drives this comes from the sun. I'm not going to go through all this again. I've talked about how the light from the sun splits water molecules into oxygen plus protons and electrons. The electrons go through the thylakoid membrane. The protons accumulate on the inside of the thylakoid membrane. And finally, this is taking us now to the next step, which we'll talk about next time, and that is respiration. And in respiration, oxidative phosphorylation occurs. That is where we get most of our energy. So energy in the form of ATP from glucose by way of respiration in not the chloroplast, but the mitochondrion. Electrons from glucose are passed through a series of reactions to inorganic molecules like oxygen. then chemiosmosis generates ATP. And you get many ATPs per cycle. Now, what is chemiosmosis? It's simply the movement of ions across a membrane down their electrochemical gradient. So as the video showed, and as I've been trying to emphasize, when the electron transport chain occurs, the electrons are transported within the membrane, and as they are, protons accumulate on one side at a higher concentration than on the other side. And because of this concentration gradient, they can pass through this ATP synthase complex to convert ADP to ATP. And this process is called chemiosmosis. So that's a term that you should remember, and remember what it is. Simply the movement of ions across a membrane generating ATP. I'll give you a preview of what we'll talk in much more detail about next time. And what this is really showing is oxidative phosphorylation. Oxidative phosphorylation starts with glycolysis. So glucose, the 6-carbon sugar, is split into 2 3-carbon sugars called pyruvic acid. And when this happens, you get 2 ATP molecules. So remember, we're back here. This is substrate level phosphorylation. You go from pyruvate converted to a product of pyruvate plus ATP. Then these 2 pyruvic acid molecules enter the Krebs cycle. It's not the Calvin cycle, it's the Krebs cycle. They're converted to 2 acetyl-CoA molecules. Carbon dioxide is given off. Krebs cycle gives off more carbon dioxide, and the Krebs cycle generates 2 more ATPs. But the real gold mine is in the generation of NADH. So at every step here, we're getting more NADH. That means electrons being transferred down the electron transport chain. And remember, when that happens, protons are accumulating on one side of it so that chemiosmosis occurs. And the final electron acceptor is oxygen, so, which, because it's accepting electrons, it converts it to water. And so the process of going through all of this generates 34 molecules of ATP, plus the 4 molecules that we generated at substrate level phosphorylation, adds up to 38 total ATP molecules from 1 molecule of glucose. In terms of ways to generate ATP, this first step is substrate level phosphorylation. The second step is also, but all the other steps are oxidative phosphorylation. That's what generates the most ATP from glucose, oxidative phosphorylation. And since I'm talking about this, I'll give you, excuse me, a preview of next time. We're going to be talking about the citric acid cycle. It's also known as the TCA cycle or the Krebs cycle. And it occurs in the mitochondrial, not the chloroplast. We're moving on from the chloroplast to the mitochondrion, which has a structure, as you see here, has an inner membrane complex called cristae, an intermembrane space, and a matrix. The electron transport chain and chemiosmosis occur in this inner mitochondrial membrane. So this arrow here is pointing at this inner membrane. So this is analogous to a thylakoid. Analogous to a thylakoid membrane in a chloroplast. Within this membrane is where electron transport occurs, protons are accumulated, and when they pass through the membrane, ATP is generated. I will go over the Krebs cycle in more detail next time. The point I want to make here, these are all the structures, so you can see how chemically complicated it is. You don't have to remember the names of these intermediates or their chemical structures. Just know that glucose feeds into pyruvate, which is converted to acetyl-CoA. That starts the Krebs cycle, and ATP is generated in it. So the function of the Krebs cycle is that it oxidizes glucose, fatty acids, and amino acids to carbon dioxide through a series of enzyme-controlled steps. And the result is that high energy electrons from the oxidation of these foodstuffs are transported by activated carriers, NADH and FADH2, to the electron transport chain. We'll review this in more detail next time, but let's just compare these two cycles that we have. The Krebs cycle, which occurs in the mitochondrion in respiration, and the Calvin cycle, which occurs in the in the chloroplast and photosynthesis. So the Krebs cycle is part of the aerobic respiration process. Calvin cycle is part of photosynthesis. Krebs cycle is in the mitochondria. Calvin is in the chloroplasts. Krebs cycle is a catabolic process. It tears molecules apart and releases energy. Calvin cycle is an anabolic process. It consumes energy to build up molecules. In the Krebs cycle, ATP and carbon dioxide are produced. In the Calvin cycle, ATP and the carbon cycle are used. Krebs cycle takes place in all aerobic organisms. The Calvin cycle takes place in plants. Calvin cycle only occurs in photosynthesis, and that only occurs if there's chlorophyll. Krebs cycle oxidizes glucose. Calvin cycle creates glucose. And finally, Krebs cycle occurs in the presence of oxygen, whereas oxygen is not involved in the Calvin cycle.
Real quick, and I don't know if I heard this incorrectly earlier, or I'm a little confused, but so photosynthesis happens when light is hitting a plant, but for the respiration process, the Krebs cycle, right? That's typically happens when it's dark.
Yes.
Okay.
Yeah. So we animals don't have to be in the light in order to get energy.
Gotcha.
Eat the food that plants produce, and we can undergo the Krebs cycle, respiration, in the dark or the light. Light is irrelevant in the Krebs cycle.
Oh, I see, because it's generating everything during photosynthesis and then we take in that energy ourselves. So we don't actually have to, I mean, that just goes for any like organism essentially, or aerobic organism.
Yeah, this is referring only to aerobic organisms.
Yeah, gotcha.
When next time we'll talk about what happens when there isn't any oxygen. It's a completely different outcome. Okay, and then I've been talking mostly today about photophosphorylation, but I've mentioned briefly oxidative phosphorylation, and this is a comparison of the two. Let's talk first about photophosphorylation. It's the process of ATP production from sunlight during photosynthesis, whereas oxidative phosphorylation produces ATP using enzymes and oxygen. So this is when we breathe and we're gaining energy from food, we're undergoing oxidative phosphorylation. Now, the energy source for photophosphorylation is sunlight. The energy source for oxidative phosphorylation is glucose. You combine glucose with molecular oxygen, generate ATP. Location, photophosphorylation occurs in the chloroplast, oxidative phosphorylation in the mitochondria. Photophosphorylation occurs during photosynthesis, whereas oxidative occurs during cellular respiration. In other words, anytime you eat food and digest it, oxidative phosphorylation is going on so that you can get energy from the food you eat. The final electron acceptor for photophosphorylation is NADP, and it becomes NADPH. In the case of oxidative phosphorylation, the final electron acceptor is oxygen. And the electrons from the electron transport chain, when combined with oxygen, make water. And it's because of this requirement for oxygen that we breathe, that we need to breathe to get oxygen as the final electron acceptor. If we don't have any oxygen, the entire process stops. So I have here at the bottom the elements of the two. Photophosphorylation starts with light and water. Light and water combined give electrons plus carbon dioxide, which then ends up with glucose and oxygen. Oxidative phosphorylation starts with glucose and oxygen, releases energy, and ends, rid of this, ends with carbon dioxide and water. And on this slide, just a summary of what I've been telling you about the different ways that a cell can produce ATP. Substrate level phosphorylation, relatively low yielding. It's a 1-for-1 ratio of glucose to ATP. Oxidative phosphorylation is very lucrative when it comes to getting a lot of ATP from glucose, and that's shown in this diagram. Start with glucose. 1 molecule of glucose generates 38 molecules of ATP, whereas in substrate level phosphorylation, you get 1, actually 2, because glucose is split into 2 pyruvate molecules. that you see here. Glucose splitting to 2 pyruvates gives you 2 ATPs. And then there is photophosphorylation, where the pigments, meaning chlorophyll and photosynthetic cells, trap light energy and then convert it to chemical energy through electron transport and chemiosmosis. And that's illustrated by this diagram. And I want to emphasize, you should know what chemiosmosis is. It's the transfer of, or I should say the movement of, this says ions, it really means protons, across the membrane. And this membrane can be either the thylakoid or the inner mitochondrial membrane. But when protons are transferred across it through this ATP complex, ATP is generated. So it's the movement of protons down their electrochemical gradient. So just one more summary slide before we quit. In photosynthesis, we have light reactions and dark reactions. They both occur in the chloroplast. Light reactions, sunlight comes in, strikes the thylakoid membrane, which splits water into protons and electrons, plus oxygen. So water goes in, oxygen comes out. But this starts a process of electron transport and proton accumulation in the thylakoid. And the result of that is chemiosmosis, where ATP is generated. ATP plus NADPH, its reduced carrier, are then transferred from the thylakoid to the stroma. Another term you should remember, the stroma, this is still within the chloroplast, and that's where the Calvin cycle occurs, within the chloroplast. Calvin cycle occurs in the dark. So for a plant, after the sun goes down, it can still keep making glucose. And the remarkable thing about the dark reaction is that there's this enzyme complex called Rubisco that can take a gas, carbon dioxide, out of the atmosphere and convert it into carbon compounds that can be metabolized by metabolic systems. So these are light independent. The Calvin cycle keeps turning as long as there's carbon dioxide and as long as there's energy coming from the light reactions. And the end result of the Calvin cycle is sugar. I think that's the last slide I have. Yes. So our time is up and I need to go promptly. Are there any questions or comments before I end the session?
No,
Did you say that all of this is going to be on the quiz on Wednesday?
I will take questions from this slide set. But remember,
Okay.
this is only a quiz on Wednesday, and so there will be probably fewer than 10 questions. They'll be fairly short, simple questions, just to sort of spot check what you've been learning about molecular genetics, enzymes, and photosynthesis.
well, we will come to class on Wednesday.
Yes, the class will only be 60 minutes because you'll have all day to take the quiz.
Okay,
But that'll be 15 minutes out of your time.
okay,
And on Wednesday, we will focus strictly on respiration.
Okay, sounds good to me. You have a good day, Professor.
Okay?
bye, everybody.
Thank
Bye.
you.
All right, I'm gonna stop sharing.
Bye.
Bye.